everybody.
Good afternoon.
Good afternoon.
Hello.
Good afternoon, everybody.
Hello. So, Gilberto, I think you have some words to say.
Yes, I will. Our words of thanking. Thank the presenters, all presenters, students and seniors, and young scientists also. Thank the organization. You're lovely people who worked so hard. But most of all, it was so beautiful, everything you did, you know. I know you did it with love. That's what amazes me. And I love that. And the third is the generous attendance of the young students, undergraduates, graduates, and so on. Thank you. You are responsible for the success of MFIM. And be prepared because the next is on the trigger, okay? We'll have it soon. Soon, I mean next year, but the soon as possible. We are working on that, okay? Thank you very much for everything. Thank you all. I have to leave before the end because I have another. I'm sorry, but it's a very important call. I have to attend, but I'll see if I stay. I want to stay. Okay, I'll come back. Jason,
Good.
thank you.
Thank you.
Yes.
Glad to be here.
So today,
I owe you a beer. I owe you a beer. First time you drink, I'll buy you a beer.
Sounds good.
Yeah, okay. Remind me.
I'll do that.
Yeah,
yeah,
okay. I'm sorry,
so
Marcela.
no problem. So today, unfortunately, is our last session from this fourth edition of My Friends MASSA. And we have online here with us, Veronica as our special guest. She is representing the student committee organization, so the committee, so she will be online with us. And then please stay with us after the talk, Jason talk, because then we will present the best, the prize of the best presentation from the student sessions, okay? And have some closure remarks of this session of My Friends MASSA. So now it's actually with a great pleasure that we have Jason Lavinder here, because Jason, he's working a lot with antibody repertoire analysis and with HepSec and also IGSec that he will talk to us today. And he also works a lot with microfluidics to perform single B-cell VH-VL native pairing of antibodies. So Jason has a PhD in biochemistry from the Ohio State University. He was also a principal scientist in Pfizer endogenous antibody discovery group from 2016 to 2018. And in 2018, he co-founded a company that calls Linked Up Bioscience, and he was also the vice president of technical development from 2018 to 2020. And now he's currently in the advisor board of this company. And he's actually now a research scientist in the Institute of Human Immunology and Protein Therapeutics at the University of Texas. So it is with a great pleasure that we have you here, Jason, to hear your work. So thanks again for accepting the invitation.
Thank you, Marcella, for the introduction. Thanks for inviting me. Let me get the slides shared here. Okay, and okay, does everybody see a slide? Does they see the title slide?
Yes.
Okay, great. So as Marcella already kind of mentioned, my background is, my PhD is in biochemistry. My background is primarily protein chemistry. So my postdoc, when I started my postdoc at UT Austin in 2009, I didn't really come in with any immunology experience nor mass spec experience. And so the work that we did in Georgiou's lab during my postdoc started with actually some NGS sequencing of B cell repertoires to understand the response to infectious disease and as well as to vaccination by looking at B cell repertoires by NGS sequencing. We quickly realized during that time that some species, like rabbits and other species as well, sheep and goats, we looked at several different species. B cell sequencing was not a great tool for antibody discovery in the sense that if you sequence, for example, bone marrow, spleen repertoires of B cells, and you then try to make those immunize the animals, that is, and you try to make the high frequency kind of expanded B cell lineages from those animals, they don't necessarily map to what you immunize the animal with. And so what we really wanted to do at the beginning of my postdoc was find a kind of a tool or a pipeline which we could use to actually identify directly from the serum of these animals the antigen-specific B cells, or sorry, antigen-specific antibodies that are circulating. And so we collaborated with the Marcotte lab down the hall from us at UT Austin. I worked with a postdoc in that lab who had a lot of mass spec experience. And then I kind of worked with him in terms of developing some of the protein chemistry. And then I worked a lot on the kind of the B cell side of things as too, because we still do a lot of B cell NGS sequencing because this is all database driven, bottom up mass spectrometry. So I'll kind of get started in the slides in terms of background, in terms of what that pipeline looks like, and kind of give you an overview of what we do. And so the primary way we think about antibody repertoires is not as one distinct repertoire, but rather as two individual repertoires. One being the actual antibodies, the endpoint protein antibodies that circulate within the blood, within the plasma. And then the second repertoire would be then the B cells that are circulating within the peripheral blood as well. Obviously, you got then bone marrow and different populations of B cells as well that are in these lymphoid tissues that also then are there. But these two repertoires, the B cell repertoire and then the plasma inner repertoire, are actually distinct. There's no reason then for them to overlap completely biologically in terms of their function. And we find that if you look at B cell diversities versus looking at plasma antibody diversities, they're very different. And so looking at both repertoires is important to really understand an immune response to an infection or a vaccine. And then really looking at the endpoint functional molecule that protects us from infection, the actual circulating antibody, was a key step forward for us really understanding what's going on in an individual when they get infected or vaccinated. And so the pipeline that we have is, again, looking at both sides. We look at on top is what we call IgSeq, which is the proteomic pipeline that we use to actually identify at a single molecule, a single antibody level, the antibodies that are circulating in the blood. The bottom part is then the B cell sequencing that we do in parallel from these same individuals to understand what the B cell repertoires look like and how they overlap with the circulating serum antibody repertoires. And so hopefully you see my cursor. I'll kind of go through the steps of the pipeline. The first thing we do is isolate IgG or IgA. We can do IgG or IgA proteomics from the plasma using protein G. We then take the purified IgG. We put it through then a column in which we have beads that are coated with whatever antigen of interest that you have. So if you're looking at, for example, a COVID response, you might put spike protein on these beads. If you're looking at an influenza response, you put hemagglutinin on these beads. And so you're purifying only the antigen-specific fraction then of the circulating IgG. You then elute that fraction. You then do a pretty standard proteomic approach where you denature, reduce, alkylate, and then trypsinize the eluate from that column. And then we're doing, again, tandem mass spectrometry bottom up, in which we actually are using databases to help us then identify the peptides that are derived from the elutions. We also then look at flow through, which would be the non-antigen specific fraction as well to compare. And then the B cell side of it, again, in parallel from the same donor, we sequence millions of B cells. These could be sorted subsets. These could be just straight PBMCs. We have a variety of different kind of, depending on the project, really. Primarily, we do a lot of PBMC sequencing. And there's kind of two parts of this pipeline. One in which is a a VH only kind of sequencing in which we're basically looking at the heavy chain only sequencing of the B cells that are derived from the blood. This provides a really deep database, deep sampling of the B cells that are circulating. So we get tens of millions of reads on those. Then we have a couple million B cells that we aliquot aside, in which we are then putting through microfluidic pipeline, in which we encapsulate single B cells into microfluidic droplets. We then perform some molecular biology magic inside of those droplets to link up the heavy and the light chain transcripts of the antibodies that these B cells are making. These are essentially displaying as a B cell receptor. And so we can pop open the B cell in the droplet, isolate the RNA into a bead, and then using, again, some molecular biology magic, some primer sets to help us link the heavy and light chain sequences up inside that droplet to give us then single B cell information of heavy and light chain pairings. And so we then do deep NGS sequencing on that amplicon as well. So it gives us a really, really deep database of the B cells that are circulating within that individual that allows us to then make identifications of the mass spec data. The sensitivity of the IgSeq is down to 10 nanograms per mil based on some spike-in experiments we've done. Again, the full-length IgG data that we get is from the BCR-seq side. So we know the heavy light chain pairings. We know the full sequence information once we identify a lot of these peptides on the IgSeq side. And then the coverage that we see based on some machine learning approaches detecting signatures of antibody peptides, we see that we can basically identify with high confidence greater than 70% of this detectable circulating IgG. And all the quantitation is based on XIC peak intensity. Hopefully there's no chat questions, because I'm not seeing the chat. I got full screen slides on right now, but feel free if there is, just to interrupt me. So primarily the IgSeq side, this top part here, the proteomics, is focused really on the heavy chain CDR3 peptide. Obviously, we get all these peptides from heavy and light chains that show up in the mass spec data, but there's essentially a level of information that is unique to the CDR3 peptide of the heavy chain. The reason that is when you look at how antibody development occurs, you get antibodies through what's called VDGA recombination. Some of you are probably familiar with this, some maybe not. VDGA recombination results in a junctional diversity in the purple here, where you get random end nucleotide addition. This D region, this region right here where the D region is really this region where the CDR3 is. And that's the region you see if you look at a sequence logo of a heavy chain repertoire from like B cell sequencing. You see that the CDR3 is very diverse. There's not really major that's using a variety of different amino acids in the CDR3. So that CDR3 peptide for the tryptic peptide from this conserved arginine, which is germline encoded right upstream of the CDR3, and then this constant region lysine, another tryptic site. This basically gives you a nice tryptic peptide of the right length to act as essentially a barcode for each antibody that's circulating in the serum. And so our quantitation data, and then pretty much the focus of the proteomic data when we actually are really trying to understand diversity of the antibody repertoire, focuses on detecting and then quantifying this CDR3 peptide that results from each antibody that's circulating. We also then again have upstream peptides that we can look at, but most of the data presented, most of the analysis you're going to see is going to be on the CDR3 peptide. So it's CDR3 spectra typing of circulating antibodies using mass spectrometry. And so the data readout typically looks like this. We have these histograms that we produce after we get the LC-MS data analyzed. We can then basically cluster based on B cell data. We can cluster these peptides that we identify into lineages. And so we'll typically present the data at a lineage level. And so we may have, for example, for this top lineage here, each row, each bar is a lineage. The x-axis then represents the relative percentage of that lineage that's circulating in the serum that is specific to the antigen. So in this case, this is a NAMP6 specific antigen specific pull down on an affinity column from a donor that's been vaccinated with that vaccine. And we see that 20% of their NAMP6 IgG has this lineage, which this is just a representative CDRH3 that is shown. Now, this lineage could have some similar CDRH3, for example, that looks similar to this CDRH3, but not exactly matching. It's the clustering basically gives us lineage level data and then we present that in a histogram format. And then we can use the B cell information to help kind of fill out some of the other information that we need, such as V gene usage and J gene usage of the antibodies, as well as somatic hypermutation levels across the full amino acid sequence. And again, we are seeing those upstream peptides as well, but the data you see in the histograms is based on the CDRH3 peptide, which is highlighted in red for each of these lineages. You can see the arginine tryptic site there that's conserved across 90 to 95% of antibodies that are circulating in the serum. And then some of these have internal tryptic sites that show up as well. So you get maybe shorter CDRH3 peptides that oftentimes map uniquely to each lineage. And so the outline of the talk today, I'm going to talk about in terms of the data that we produce using this pipeline. I'll talk about a recently published, well, last year, somewhat recently published article in Science where we looked at natural infection for SARS-CoV-2. We were able to identify a convergent public class of antibodies across our donors that are specific to the NTD domain. And then if I have time, hopefully I can go quick enough where I can get into some of the data on RH5.1, which is an experimental malaria vaccine. This is collaboration with the Draper Lab at the Jenner Vaccine Institute at Oxford. It's very exciting data, so I wanted to kind of start talking about that today as well if I have a chance. And in that project, we're actually able to map both in the plasma, antibodies circulating in the plasma of both neutralizing as well as synergistic or potentially non-neutralizing antibodies that are present at high levels as well. And I'll explain what that means as I get to it later. So again, starting with this SARS-CoV-2 project, we had 4 donors that we focused on based on titers that were from low to medium titers all the way up to high titers. We kind of spanned the titer range for the 4 that we chose. This is, again, the founder virus. This is the original Wuhan virus that they're infected with. Again, mild, moderate disease. And it was not hospitalized patients. Again, a range of titers. And so this is samples that were taken approximately a week or 2 after the post-symptom onset after initial infection. And again, we use this affinity column to purify the response from the serum, the IgG response, using either the full spike domain, the spike extracellular domain, spike ECD, or we also then purified against the RBD only domain as well in parallel to look at the RBD specific repertoire as well and see how that overlaps with the full spike repertoire. So this is just an example donor I'm showing here. It's a very polarized donor actually compared to some of the other donors we looked at, where we only had 6 lineages that were identified above 0.5%. So there are some very low abundance lineages that I'm not showing, but for the purposes of just looking at the data, we're looking here at any lineage that shows up above 0.5% against the spike protein, again, in the plasma as an IgG. And so what you see on the right is the spike ECD full spike data. On the left in black is the RBD specific lineages that are from a separate RBD pull down. And so what you see right away is in terms of abundant antibodies, we only really have one major RBD abundant antibody. That's one major antibody that binds to the RBD. The CM32 lineage here that's circled in green, that's highlighted in green. Again, binding to the RBD. This is just a illustration of kind of which domains these are binding to. And again, it represents 95% of the RBD specific pull down, which is consistent then with the fact that it's the only major RBD antibody that's pulled down in the full spike data. And so a very polarized repertoire, not very diverse in the sense that there's only one major RBD binding antibody in this donor shortly after infection. And then you see there's this antibody repertoire is dominated really by S2 directed antibody. Lineage 1 is approximately 50% of the repertoire, the IgG repertoire in the plasma that's against the spike. And so a dominant S2 directed antibody that is present. And then we also then have 2 NTD directed antibodies as well that are high frequency, 15 to 20%. So a very polarized plasma repertoire in this donor. A very interesting response compared to some of the other types of responses we've seen with influenza, which we may get a very, very diverse response to the serum. This is very polarized. And so looking at other donors, we do see that not all donors are polarized like the donor I just showed you. Another donor, when looking at their plasma antibody repertoire, has around 15 to 20 lineages. Looks like 15 lineages that are present. These are 2 different time points, light blue at day 12, dark blue at day 56 post onset of symptoms, a lot of overlap, not too surprising. Not a lot of changes between those 2 time points. But again, what you're seeing, if you look at it ordered by that later time point, day 56, the top 4 lineages are against the S2 domain. And so again, the repertoire in the plasma with natural infection does appear to be dominated by non-RBD lineages. With, if you look at all 4 donors, this kind of pie graph now show all 4 donors together, an average of 16% of the IgSeq, the proteomically identified lineages against the spike protein, are against the RBD, whereas 84% on average are against non-RBD epitopes. And this shows you the individual donor kind of breakdown. And so again, just to summarize, plasma repertoire IgG lineage diversity and across the 4 donors is pretty polarized, 6 to 22 lineages in terms of above 0.5%. There's some very low abundance lineages as well that I'm not really showing in these histograms. The top lineage in every donor is an S2 directed antibody. So this appears to be a trend, at least we're seeing across these 4 donors, but is also consistent then with a lot of studies that are coming out that are looking at B cell repertoires. That if you look, for example, at this particular study, looking at greater than 2,000 MABs, again, natural infection. This was a 2021 paper. So an early kind of paper that was looking at natural infection. The majority of those antibodies bind outside the RBD when you make them. It appears that at least with infection, we get a very strong response against these non-RBD epitopes. The other major finding of this study is, let me go back to the slide real quick. You can see these 2 red antibodies that I have illustrated here. Again, these are anti-NTD MAB lineages. I've kind of labeled them with a number, CM17 and CM25. We see these NTD specific lineages showing up at decent abundance in all 4 donors. And what's interesting is they almost always have this IgHV124 germline heavy chain antibody, germline gene that's used. So it kind of struck us pretty quickly that maybe this IgHV124 germline is being used as a public response, a common or shared response to the infection across donors, which would be a very unique finding. Usually antibody repertoires between donors are very unique. Public responses are known to exist in other types of challenges, infections, vaccines, but they're not common. And so when we saw that the NTD-specific antibodies often use this IgHV124 germline, we decided, okay, let's see how shared this is structurally. So we saw the structure of this CM25 lineage. This is an antibody that was from this lineage right here. And this structure showed us basically that it bound to this epitope on the NTD that involved residues in the N3 and the N5 loop of the NTD. The CDRH3, the VH contributed 89% of the interfacial area. The light chain had barely anything to do with this interaction. It's mostly heavy chain derived, which makes sense in terms of the public response, the heavy chain being the way IgHV124 pretty consistently. The light chain was variable across those antibody lineages. And then you can see that 35% of the surface area is by the CDRH3 loop. We saw this conserved allyl dipeptide that was conserved as well in the CDRH3 in these public antibodies, and that kind of buried itself into this cleft between the N3 and the N5 loop. What was exciting is that shortly after we published this in bioRxiv, there was some articles that came out that supported our finding, that was finding additional IgHV124 NTD-specific antibodies that were, again, these are neutralizing antibodies. These are protective antibodies that were in other donors when looking at B cells. They're at high frequency in other donors. So it supported our finding that this is a public response and that it's public response that is actually high frequency within the serum. And so this public response also appeared to be driving a common immune pressure in naturally infected donors, in which you see mutations that were pretty readily found across variants that were in the N3 and the N5 loops of the NTD, showing that the virus was actually avoiding this public response by mutating these loops so that these common antibodies that had this common epitope could not bind anymore. And so, forgot to mention also, the structural overlap of this CM25 antibody was consistent with the structures in terms of binding the N3 and N5 loop. The residues that were used for contact in the IgHV124 germline were consistent with the structures that were published around the same time from other groups. And that's how we kind of knew from a structural standpoint that this was a public response as well. the mutation data then backed up the fact that these at a population level, the virus was trying to avoid this neutralizing antibody response that was public. Okay, see what my timing is here. I think I should have time to talk about the second part of the talk, in which I'll present some data on an experimental malaria vaccine. This is in collaboration with the Jenner Vaccine Institute. And so just to give you a bit of background, the malaria life cycle is really complex. It involves 3 parts. The first part for the life cycle is this kind of the initial infection, in which a mosquito bite infects a donor with these sporozoites from the saliva of the mosquito, gets into the blood, essentially is able to float along to the liver through the blood, infect hepatocytes in the liver. These sporozoites then, after about a week, will form these vesicles. I forget the name of these vesicles. I'm not a malaria expert. I forget the name of these vesicles, but essentially these vesicles are full of what's called the merozoite, which is the second stage of the life cycle here. The merozoites are released and quickly are able to then infect red blood cells. And so we have essentially what's called a pre-erythrocytic stage of malaria, where sporozoites infect the liver very quickly. And then we have this blood stage part of the life cycle of malaria, in which these merozoites infect the red blood cells and then essentially use the red blood cells as factories to produce more of themselves, which then through a cascading kind of set of infections or invasions cause a massive level of merozoites in the blood that cause the symptomatic the fatal condition oftentimes with malaria. These merozoites essentially overwhelm the red blood cells. And so the third part of the life cycle is a sexual part of the life cycle involving gametocytes. There is some vaccines that's focused on that's called transmission blocking vaccines as well. I won't talk about that today. Primarily, the vaccines that are really being developed are looking at either this first stage, and that's the recently approved RTSS vaccine, as well as an improved version of that called R21 that's currently in phase 3. And so these two are looking again targeting the pre-erythrocytic stage. But today I actually want to talk about a blood stage experimental malaria vaccine. Again, Simon Draper's our collaborator in this project at the Jenner Vaccine Institute. He's been working on these blood stage malaria vaccine for a long time now. And this one in particular, the target is RH5 protein, which is on the surface of these merozoites. This in particular, this version is called RH5.1 vaccine. And so what is RH5? What is this protein? Again, it's on the surface of the merozoite in the form of this trimeric complex involving this Plasmodium falciparum Ripper protein, as well as then the CIRPA protein, and then again in yellow, this RH5 protein. The RH5 protein in this complex is what makes direct contact then with the receptor on the red blood cell. That's called basogen. This is the very important part of this. That's the reason it's so promising as a blood stage target for malaria vaccination is that I just lost my cursor. There it is. RH5 has very limited polymorphism, which is a major problem with malaria vaccines for some targets, in which cross different strains of malaria, across basically as the parasite mutates, it's able to mutate around essentially the vaccine. But RH5 is known to have very limited levels of polymorphism, which makes it a strong candidate. It's also non-redundant in terms of invasion pathways. This interaction with basogen is absolutely required for invasion, and so there's not really any way for the parasite to mutate around this very easily. And so I want to talk about initially some of the single B cell data that's out of Oxford, and then I'll get to some of the proteomic data that we've discovered as well. The single B cell data kind of shows you this initial data will help you understand kind of the different epitopes in the RH5 that are being targeted by antibodies. There are essentially, the RH5 is like a diamond looking, or I guess kite looking structure, alpha helical. The N terminus of the RH5 is linear, it's just a disordered region. Then there's this disordered intrinsic loop as well down here at the bottom. And then really not shown very well is there's a C terminal. It's not really noted on the slide, a C terminal linear epitope as well with RH5. But then you have obviously a lot of these conformational epitopes on the protein that are being targeted by antibodies. And if you look at the single B cell data that was initially derived by Oxford, characterizing the response to RH5 by antibodies, the majority of the antibodies they found were against these conformational epitopes here in this kind of orange, this light orange. whereas only a small portion, this dark brown orange, were to these disordered regions. That was an important thing to note because the fact that most of those antibodies targeting these disordered regions are non-neutralizing, and so potentially not really all that useful. Of these antibodies targeting the conformational regions of the protein, a portion of them targeted the basogen binding site up to the top of this kind of diamond-looking structure. Essentially, they call it the diamond tip of the RH5. And so those were the most potent neutralizing mAbs that they found targeted. And then about half of the antibodies that were conformational targeted kind of the non-diamond, the bottom half of the structure. However, some of them did neutralize, and so it was interesting to really find out how are these neutralizing, as the epitopes obviously were not known. They clearly were not competing directly with basogen. And so the initial data to really understand epitope coverage on the RH5 was looking at a viral vector RH5. This is, again, data from Oxford. And you can see with the 100, 200 or so antibodies that were made from this trial, the binning data here. This was generated by Cartera, essentially a high throughput SPR, looking at competition of the antibodies with each other. And so if an antibody compete with each other, essentially you would see it in the Cartera data. And then on the y-axis, the readout, when you look at each of the bins, you can see the binning of the Cartera. But then the y-axis shows a readout of a functional assay looking at growth inhibition, the growth inhibition assay. And what you can see pretty clearly is that this bin number 1, these antibodies that compete with each other here in bin number 1, which if you look at the structure is on the top part of that diamond, as one might expect, where that basogen binding site is, has a very good functional activity with a very low EC30. You then see bin 2 and bin 3 also. Some of them have pretty good functional activity, whereas 1 or 2 of them do not. And again, those epitopes are in blue here and then in brown. And so all 3 of these bins that show functional activity, at least some mAbs with functional activity, are on the top part of that diamond structure where basogen interacts. And not surprisingly, some of these other ones that are on the bottom are not showing neutralizing activity. What's interesting is, well, actually, I'll get to that in a slide or two. This R5.011 has some interesting features to it. Although it was non-neutralizing, did show some interesting functional effects. And so I kind of want to point that out now, just so you'll remember it. This again is mapping to a bin that is on the bottom part of this diamond shape and has demonstrates negligible GIA. Just keep that in your memory. So vaccine regimen for the samples that we received at UT Austin were from a new trial that was VAC063 trial, where instead of using viral vector, they are now turning to a subunit or a protein vaccine here. It's a RH5.1 with AS01B adjuvant. There was essentially this 3-dose regimen, 1 month apart, that was used for immunization of UK adults. We focused on 5 UK adults. So it's based on our throughput. We can't do hundreds, so we used titers to really help us focus on picking 5 samples out. We're at this G5 time point, and that's the data I'll talk about today. We're also then looking at some samples from after a 4th dose 4 months later at the G7 time point. I don't have data yet on that, but that is in the pipeline. And so these first 2 donors I'm going to show, this IgG histogram is actually looking at not just the top 0.5%. This actually shows you every detectable lineage that's circulating in the plasma against RH5. And so again, the y-axis is the plasma IgG, again, relative percent abundance in terms of RH5 specific antibody that's pulled down in an affinity column and then shot on the mass spec. Again, using CDRH3 spectra typing, we see there's almost 1,000 lineages that are being detected. Some of them very low levels, whereas some of them, again, if we were to look at 0.5% cutoff, which actually I'll show you in a slide or two to look at the top part of this histogram, there's about 20 or so that are pretty frequent in terms of their abundance. So this is donor 29 up top, donor 36 on the bottom, 2 different donors, very diverse repertoires that are being elicited by RH5 vaccination. In green, I highlighted these lineages here that are actually showing up on the Oxford side in terms of their B cell sequencing. And so what they do is they actually take the RH5, they label B cells with it. So you have antigen-specific labeling of B cells, and they sort those out by FACS. And then the question is the B cells that they sort out by FACS actually show up as antibody in the serum? Are these B cells they find, these memory B cells that are specific to the antigen, actually have corresponding circulating plasma antibody? Some of them do, some of them don't. 3 of the 9 lineages that they derive by B cell antigen-specific sorting we find in the serum for donor 29, and in donor 36, half of them, 11 of 22, show up in the serum, whereas the other half do not. So it's an interesting comparison. Again, I noted early on, the antibody repertoire is both, it's kind of 2 different repertoires. So it's an interesting comparison to look at the 2 different kind of compartments and see how they overlap. And so the question then is the plasma, is it enriched for neutralizing antibodies? Is it enriched for non-neutralizing antibodies? Interestingly enough, when we look at the proteomic identified plasma antibodies, these are just the first kind of MABs off the pipeline. So each data point here is a lineage, a MAB derived from a lineage that's in the plasma. Over here on the right in blue is all of the MABs that were derived from the antigen-specific B cell cloning. And these are all functionally now annotated for how good are they at actually blocking infection. And what you can see is if you look at the plasma MABs versus the B cell derived MABs, it does appear actually that there is a lot more non-neutralizing antibodies in the plasma at an endpoint level than compared to the B cell data. And so that's very obviously interesting. And the question is, why is that? But also then another question is, are these non-neutralizing antibodies, these non-protective antibodies, are they beneficial? What do they do? And so what was interesting from another publication from Oxford from this earlier data with B cell sequencing of antigen-specific B cells, B cell cloning as well, is that they actually found some MABs. I noted this earlier on in that slide and told you to remember this R5.011. They found some non-neutralizing MABs that actually, when you combine it with a neutralizing MAB, this RA5.016, for example, is a really potently neutralizing MAB. They found some other ones that were kind of mildly neutralizing, and then other ones that were negative. When you combine it with these other MABs, it actually synergizes or potentiates their functional activity. So by itself, again, I showed you earlier on, 5.011 doesn't have any neutralization potential. It doesn't do anything. It's non-neutralizing. But when you combine it in an experiment with some of these other MABs that do neutralize, it actually improves upon their neutralization potential. You can see, for example, here by itself, 5.017 has a GIA percent, so 70% prevented infection here. Whereas when you combine it with this potentiating 5.011 MAB, all of a sudden it is preventing 100% of infection in this assay. And you can see correspondingly then with some of the GIA low MABs, these 007, this 015, which has a very low level of, barely detectable level of neutralization activity for some of them, for example, the one in purple. All of a sudden, you use 5.11 in combination, and it's up to 50%. So it appears that this particular epitope, that 5.011 is binding to, is doing something. It's not neutralizing epitope, but it's doing something to actually potentiate other neutralizing antibodies that bind to other epitopes. And so the question then in the study was, why? How does that work? And you can see in this data here, again, out of Oxford, that the non-neutralizing MABs, this 5.011, when you look at actual individual and kind of invasion at a single cell level, what it does is actually it slows down the invasion of the merozoite into the red blood cell. So when 5.011 is present, it takes longer for the merozoite to actually invade the red blood cell. That doesn't do anything by itself in terms of neutralizing. Eventually, it does invade the red blood cell. But what happens is it slows it down enough that some of these neutralizing antibodies actually work better. And the reason for that is because the invasion times are so short, they're so quick that unless you slow it down, it's tough for the antibodies essentially to bind to the RH5 and prevent infection in that short time scale. And so the question that we had is, do we see those in the plasma? Do we see these potentiating antibodies in the plasma? Obviously, 5.011 was a B cell derived antibody that was found at Oxford. We wanted to know in our donors, do we see these same antibodies and at what level? What we see initially is that when you look at these two donors, donor 29 and donor 36, I didn't label the y-axis, but again, this is percent abundance of RH5 specific IgG in the plasma. The top lineage in green here that we know we have data on from the B cell cloning side is actually a potentiating antibody. This isn't 5.011. This is another antibody that's similar to it that's found in each of these donors. And as far as the B cells that were derived from these donors and how they overlapped with our serum, the top one that's found is actually, fortuitously enough, a potentiating antibody. So that got us off to thinking, how many more of these are there? So we had all these other antibodies to look at. The ones in asterisk are essentially ones where we had, again, B cell data, full-length B cell data that we could use for recombinant mAb production. So we could make all these antibodies in the lab and test them. So that was kind of the next step. And so those were produced and expressed. And really, at that point, so we're focused here in the red box on just donor 36 for this next set of data I'll show you. So initial screening is like where on the RH5 are these donor 36 antibodies that are circulating in the plasma binding. So we had different antigens for initial screens, full length as well as denatured full length. We then have just a N-terminus piece that we're screening in ELISA against to see if we can quickly define N-terminus specific antibodies, as well as then another engineered RH5 that lacks the disordered regions. So that intrinsic loop is gone, the N-terminus is gone, the C-terminus is gone. And then what we see is here another kind of antigen where it's just the top part of this diamond here expressed by itself. And that helps us to identify antibodies that bind to the epitope that's kind of the potently neutralizing epitope. And so for all the antibodies we made from this donor 36 that are these high abundant circulating plasma antibodies, we're able to map them under these different antigens, these different kind of antigen domains. And we can see basically that a large percentage of them are actually binding to these linear epitopes, which is unlike some of the B cell data I showed you earlier. When you look at all the antibodies that are produced from B cells, we have a lot of antibodies in the serum that are binding to the disordered N-terminus, as well as in the disordered intrinsic loop in blue, as well as then a lot of antibodies that are binding to that kind of trimeric, that full RH5 region, but it's actually linear epitopes within that region. This could be a C-terminal. Some of these could be C-terminus antibodies, directed antibodies. We haven't really clearly defined that yet. But then you have about 29% that are binding to conformational epitopes in the RH5, and another 6% that actually bind to that bundle protein. So these are based on abundance in the serum, these percentages. And that you can see it's like just one antibody, one lineage is 5.077. These are ordered on rank. So this would be the 7th ranked lineage that binds to that bundle. And so to get really more into detail on the binning, on the exact epitopes, Oxford performed binning using Cartera of all of these antibodies, and also had some controls based on their previous data, some known controls. They already know where these antibodies bind from the B cell data. And I'm going to skip to a summary slide rather than try and deconvolute this. I'll give you the summary. And so this is the last data slide. So hopefully I'm on time, probably not. And so what you're seeing here, when you look at actually exact epitopes of these donor 36 specific plasma antibodies, these RH5 specific antibodies, we have a lot of them that are binding to this bin1, this potently neutralizing bin. So the ones that are conformational that I showed earlier, that's conformational antibodies, a lot of them are binding to this bin1 epitope, overlapping with this potently neutralizing R5.016 control, for example. And so these we expect to have probably potent neutralization activity. So that's a good sign in terms of the vaccine. There's some antibodies that are also showing up against bins 4 and 5, as well as bin 7, which are non-neutralizing bins. But if you remember, this bin 5 is R5.011. This is the bin where we found this synergistic, this potentiating mAb earlier on. And so we do see at least one, in addition to the one we talked about just a second ago, that was a B cell derived one. We find another one in the serum that's likely mapping to the same epitope that we would expect also then to be potentiating. And so we're looking at that right now. I don't have data back on that yet. But basically you see there's also some new bins that are defined that weren't previously defined by the B cell sequencing, the B cell data. And so we're now just getting a picture and a comparison of the response to this vaccine in the plasma in terms of RH5 specific IgG in the plasma as compared to what we see at the B cell level. And so I just wanted to acknowledge all of our everybody that worked on the project at UT Austin, in particular Greg Ippolito, who is my partner in crime on the antibody side of the lab. We basically are the ones that are kind of dictating these projects and kind of mentoring the grad students and the postdocs. And then George, both Greg and I, as well as Everett, who's another PI in the lab that works in the enzyme side of the lab. We all work under the umbrella of George, and so we are very thankful for all of his advice and help. And then collaborations, Dr. Jason McClellan here at UT, and who provided a lot of expertise when it came to the COVID work, as well as then Ilya Finkelstein provided some of the competition data for the COVID work, looking at epitopes. And then this is a bunch of other grad students and postdocs in the lab that played big roles in these projects. I won't go through each and every one. We have a lot of collaborators as well. I've talked about Oxford team, Dr. Simon Draper, Jordan and Kirsty have been key for that project. And then UNC Chapel Hill, big collaborator for our work on the COVID project, Dr. Ralph Barrick. PATH MVI is the malaria work. They were facilitated and did their administrative kind of facilitators for that work. And then our core facilities, the CDC, helped us out with some of the COVID sequencing. We're very thankful for that. And hopefully I didn't run too much over. I'm happy to take questions. I'll check the chat because I do not yet see it at the moment.
Thank you very much, Jason, for the great talk. I think it's a great way to apply proteomics. That's so comprehensive, and it's amazing data. I think we have, I don't know if you see already in the screen, Lisa asking.
Okay, I'll start there. Great. Let's see. Oh, yeah, so that's a good question. So it's actually common in the sense that it's germline. It's a germline encoded antibody, or the mutation level is very low. So it's easy basically for example, if you get infected, you don't need to have a lot of mutations to form a polyclonal type antibody to actually binds and neutralizes the spike. So it's common in that sense. It's easy to actually have that response, and I think it's the reason we see it as a shared response. On the other hand, IGHV124 is normally a rare germline. And so if we were to look at BCR sequencing in a naive donor control, it's a rarely used germline. And so the fact that we see it at high levels in the plasma, and then as far as the BCR sequencing, you do see it at higher levels, but it's still like, maybe 2 to 3% of the BCR-seq data in terms of IGHV124 usage. And so we didn't really make a bunch of BCR-seq representatives to see if they are actually part of that neutralizing response. Likely many of them are, because normally IGHV124 is very low percentage. But again, only a portion were present in the serum. I'm gonna try to keep going down the list here. In your data, in general, the circulating antibodies are in the top clones in the BCR-seq. Again, yeah, they're elevated, not top. There's a lot of the plasma antibodies that show up don't necessarily appear as the tip-top clones in the BCR-seq data. A lot of times they're high read, but then there's other ones that are like tens of thousands of reads that don't show up in the plasma. And so what that means is hard to distinguish sometimes. Some of those may be something else entirely different. Some of them may be antigen specific to whatever you were infected with, but just may not be in the plasma. We had some early data from tetanus where we actually looked at that. And there is a lot of plasma blasts that are specific to tetanus that show up shortly after vaccination. And we confirm they bind to the virus, the toxin, sorry. We've confirmed they bind to the toxin, but yet don't show up in the plasma at all. So,
but if I can just complete this question of Lisa, because so then when you select the antibodies to express and study more, you only focus mainly on the circulated that you identify in the proteomics.
Yes. When we go to make antibodies, at this point for most projects, we don't make antibodies that are not present, that aren't detected. In other words, sometimes we have this antigen-specific FACS data where we might have a lot of B cells that we know bind to the antigen based on that data. They don't show up in the serum, though. So we can make some of those and confirm and look at their affinities. We've done that before, but a lot of times And then these projects in particular with the COVID work, we didn't really do that. And so we don't know necessarily through like confirmed recombinant MABs, the characteristics or the properties of those antibodies compared to the plasma antibodies that we did make. We did that for some projects. Tetanus project was one of the first projects where we really did that. But routinely, no, we don't. We just focused on those that show up in the plasma.
Circulated. Okay,
Yeah.
thank you.
How much overlap do you observe on IG-seq and RNA-seq? Yeah, similar question. Yeah, it's small actually. The BCR-seq database is huge. So you have a lot of because it's not just antigen-specific BCR-seq. It's total PBMCs or total. You can sort plasmablasts or sort memory B cells, and you can look at total populations, and that's what we typically do. We don't antigen sort everything. The malaria project, we have collaborators that did that for us, so we could start comparing those things more routinely, I guess, more easily. But the total BCR-seq database that we have is usually everything, whether it be specific to the antigen or not, and it's very diverse. The memory B cell repertoire is very diverse, so it's like a fraction of that where you see plasma antibodies specific to your antigen that you just vaccinated or just infected with that map to that. Did the neutralizing antibodies versus non-neutralizing antibodies in the malaria study have the same IgG subclasses? Maybe they have different effector functions. This is a very good question. We're trying to look into this. In terms of FC effector function, IgG subclass, it's hard for us at a proteomic level to get a lot of good information on that because of the fact this is bottom-up proteomics. The constant region is no longer that is no longer kind of linked to then the CDRH3 peptide that you identify. So you CDRH3 peptide could have came from an IgG1, an IgG2, an IgG3 for it. We don't know. And so if the B cell data where we actually do sequencing has that information, then we can start we have it then. But our BCR-seq as it currently stands, actually the primer is right on the other side of the variable region, on the IgG constant region. So we aren't far enough upstream in the IgG constant region to clearly define subclass by the BCR-seq data either. So we want to start doing that. We want to start actually sequencing further into the constant region. That way our database has all that information about subclass. But up to this point, we have not incorporated it as part of our pipeline. Greg is very, very interested and keen on doing that. It's just that the length of the read is longer, and so we started early on just trying to keep the length of the BCR-seq read for that B gene read shorter. And that also then for the pairing data makes it's pretty much you don't really have a choice. You have to kind of minimize the VHVL pairing data so you don't really have constant region information there either. So yeah, hopefully that answered your question.
So Marcelo, may I ask a question?
Of course.
So thank you very much. Really, really nice talk. I'm not an immunologist guy, so it may be very, very basic question, but I've seen that as far as I can understand, the CD3 is the most interesting part because it's the most variable one, right? In the
heavy chain, yeah, of the heavy chain.
And it's, as far as understood, it's always flanking arginine or lysine, right?
Almost always. So I did like a lot of these in silico tryptic digests early on with different species like humans and rabbits, just to understand, like if you look at BCR-seq data, take a bunch of B cells and sequence them and use that as like a database and understanding like this is somewhat relative to what we might expect in the serum in each species. Obviously it's not one-to-one, but if you look at the BCR-seq data and you're in silico digest that with trypsin, I think it's like 92% of heavy chains will have that CDR3 popped out by trypsin. And so there is a percentage, albeit maybe, I don't know if you call that small, several percent, where trypsin may not give you the right size peptide to see in the bottom-up proteomics. And so therefore you would miss it most likely. Yeah, so it's not, I can't say it's 100% comprehensive. Yeah, we looked at alternative proteases besides trypsin to see if we can kind of catch those other several percent. Trypsin's just so good though at being at the specificity and just how effective it is at actually cleaving these antibodies and these. It just that data looks so much cleaner than an alternative protease. All the different alternative proteases that we looked at. So we stuck with trypsin, realizing that there's at least a fraction that we might be missing.
Okay.
Yeah.
So then in your workflow, you don't do the nucleotide sequencing first to see which residues are there, and then use trypsin. You just use trypsin straight away.
We always use trypsin,
Okay.
except early on when we were trying to see if we could use other proteases successfully. Now we just use trypsin. We are looking, actually, it's an interesting question now for us, because we're actually looking at some species that don't have a tryptic site in the constant region. Ferret is actually one of them. Ferret is a model for influenza challenge, and ferret does not have a lysine in the constant region. This, like in the human, it's ASTK is the first 4 amino acids of the constant region. You don't find a tryptic site in ferret until like 40 or 50 amino acids in to the constant region. So we can't use trypsin for ferret proteomics. So now we have to turn to alternative proteases, and that's it's probably going to be a little more difficult, because early work just showed that trypsin was so much better. So
yeah, actually, we have observed for horse that we are studying that they have a lot. Some of the CDR3, they have a lot of lysine or arginine in the CDR3, so it is get a little bit tricky that to get this. So definitely, it's
and in that case, you might only get a piece of it.
Yes.
And if it's a small piece of it, then it isn't necessarily informative enough to tell you where it came from. So that happens to a certain extent as well. It just depends on the antibody. Yeah.
So
what about top down? Are you considering maybe to go to middle down or top down?
We've tried. We've done a little bit of exploration on that. I think others do it better than us. To be honest, there's some other groups out there that have done top down. Top down, I think, would be a great complement to the bottom up. Top down by itself is not great for identifying, obviously. I think you can maybe use it for, I don't think it's very easy to use that for identifying, though. But top down is more of a way of profiling, in my opinion. So, you know, it kind of gives you like the number of species that are there. You may not know what each species is, each antibody kind of that shows up. You may not have an exact sequence, and therefore you can't really make it and validate it or test properties of it. But it does help you get a profile of the response in terms of antibodies that are present in the serum against that antigen. But the bottom up then obviously is much better for the actual antibody identification, protein identification, for obvious reasons. So I think it'd be a great complement to do both on the same samples. We've just not seen a lot of success in our hands, and then we're not probably the best mass spec people, so there's others that do better at it. And I've seen some others, that publish really good work on that.
Nice. I think we have one more question from the chat.
Do you get heavy chain, light chain pairing information from the IgSeq? No, we do not. We have to rely upon the microfluidics that I talked about early on, kind of glossed over it a little bit, didn't talk about it a lot. That single B cell microfluidics is really the only thing where we get light chain pairing information. So that database, that heavy chain, light chain linked database, is necessary for us to actually make lineages. You saw in the one figure I had the asterisks that were applied. Those asterisks indicated lineages where we had linked information, where we can make the antibody. If we didn't have the linked information, all we can do is say, well, we identified the heavy chain in the plasma. We know it's there, but we don't know the light chain that partners with it, so we really can't make it. We'd have to do like phage display for the right light chain or something, which you can do. It's just an added on kind of thing that you'd have to do then to get to the antibodies.
Okay, nice.
All right. Thanks a lot. Those are good questions.
I'd like to thank you a lot for your talk. I will have more questions, but I can send you later. Then we can talk more about it. So thank you again for accept this presentation, and it was amazing to have you here and hear about your work.
Yeah, thank you for the invite. Appreciate it.
Thank you.
Thank you very much. Nice to meet you, Jason.
Yeah.
Same to you.
So now we will get following the session. where I can present, Tiago.
Yes.
Just
Where is it?
I am sharing already.
Oh, sorry. Yes.
Okay. So I'd like to thank you again from the Student Organizing Committee for all the work, the amazing work that they do in the backstage. So they help a lot with the divulgation and the organization of my friends MASSA, check the people that apply and also the works. So thank you a lot for you guys. And then I will pass the word for Veronica, that is here representing this committee. And Veronica is with you.
Okay, thank you a lot. And I'd like to say thank you for this opportunity to participate in this committee. And before to present the winner of the student session, I would like to say thank you for the whole evaluation committee that includes several professors from CT vaccine, such as Dra. Diana Gomes Mendonça, Dra. Juliana Silva Cassoni from the Universidade Federal do Pará, Dr. Vitor Corasola Carregari from Unicamp, and Dr. William de Castro Borges from Universidade Federal de Ouro Preto, Dr. Graziela Elisa Honsen from USP, Dr. Marcela Melo Braga, and Dr. Wagner Fontes from Universidade de Brasília. So now I'd like to announce the winner of this student section. So congratulations to Milan Avila Gleisen from Fiocruz, Paraná, that present the great study about toward confident large-scale interactomics with SCOUT. So congratulations, Milan. You will receive the Peaks of Science. So thank you very much.
And I don't know if you hear me or...
Yes, we can.
Okay.
Yes, we are.
And then it's just another comment. And congratulations, Milan. Actually, congratulations to all the students. It was a very tough work. It was a lot of very good work. So congratulations. We will get in contact with you later to ask you some informations, okay? And also give you the certificate. And then let's remind, please don't apply to go to the Ibero-Americano Conference of Mass Spectrometry that it will be in Rio in December. They extended the abstract submission until, now I need help in the English, until dia 30 de outubro. Tiago?
No, just Thursday.
Okay.
It's,
yeah. So, and please send your abstract, and we will have a special session from the BIA-PROT, that it will be the BIA-PROT Young Investigator, where we will give the prize of Prêmio Gilberto Dumont para o jovem pesquisador, que vai ser, né, selecionado, se eu não me engano, são 6 das 6 melhores apresentações lá no dia, tá? Queria agradecer mais uma vez a todos vocês por participar, a Grazi pela...
I think I took Marcela out. Sorry, Marcela. I kicked her out. Sorry.
Okay. Okay.
Você então, Grazi.
Yeah. Pessoal, só relembrando, nós vamos enviar também um formulário de avaliação do My Friends Massa, então, hoje ou amanhã, por favor, preencham lá, coloquem sugestões, o que vocês acharam, o que gostaram, não gostaram, é muito importante para nós. E, eu acho que é isso. Eu gostaria de agradecer muito a todos que assistiram às palestras, a todo o pessoal que participou da organização, os estudantes, as pessoas que participaram, que assistiram, e dizer que o ano que vem tem mais. Se inscrevam para o congresso, em dezembro estaremos todos lá presencial, que é muito mais legal, né? Então, vamos para o congresso em dezembro, a primeira oportunidade depois da pandemia presencial aqui. A Marcela está de volta. E, por favor, não deixem de preencher a avaliação, que é importante para nós saber como que nós podemos tentar melhorar. Bom, e agradeço a todos. Marcela, você quer falar mais alguma coisa?
Não, era isso, agradecer mesmo a participação de todo mundo, ficarem atentos, porque vai ser muito bom, na verdade, encontrar com as pessoas pessoalmente, né, no Ibero. Então, vai ser uma oportunidade muito boa, principalmente depois de uma pandemia, né, para conhecer realmente nossos amigos de proteômica, né? Então, é isso, não deixem de participar. E aí o Tiago quer falar, e obrigada.
Eu só queria agradecer demais em nome, né, dos demais organizadores, a Grazi e a Marcela, por terem, né, sido as hosts desse quarta edição. Acho que vocês fizeram um trabalho fenomenal. Acho que muitíssimo obrigado por terem escolhido, né, a lista de palestrantes. Acho que todos foram de super alto nível. E foi muito bom também ver, né, escutar um pouco do trabalho de vocês. E é isso, agradecer demais. Foi uma primeira experiência, eu acho que foi ótimo, né? E com certeza vamos continuar para a próxima. Então já fica aí o convite para a sociedade, que ano que vem, com certeza eu vou encher o saco de algum de vocês aí, para vocês também participarem, né, como novos editores da 5ª edição, que certamente vai ocorrer ano que vem, porque eu acho que já é um sucesso, né? O My Friends Massa tá aí já no calendário da Sociedade Brasileira de Proteômica, né? E muitíssimo obrigado para vocês duas. De novo, agradecer demais a comissão dos estudantes, né, em nome aí da Verônica. Vocês facilitam demais a vida da gente. Acho que a Marcela e a Grazi viram, né, como é que fica muito mais fácil com a ajuda delas, de todos eles, né?
Nós agradecemos a oportunidade de participar, viu? É um prazer.
Da nossa parte também, Tiago, eu acho que eu e a Marcela, né, Marcela, estou falando por nós duas, mas foi uma experiência muito legal, a gente só tem a agradecer.
Sim, obrigada, né, por indicarem o nosso nome aí para essa quarta edição, foi muito bacana, e é isso, aguardando já ansiosa para a 5ª edição.
É isso. Tá bom, gente.
Boa tarde, pessoal.
E até dezembro, né, lá no Rio.
Nos vemos em dezembro.
Nos vemos no Rio.
Até.
Tchau, tchau.
Tchau, tchau.